Hi class. In this video we're going to talk about lipids. Now lipids are defined by their solubility in nonpolar solvents rather than by chemical structure. There are a great many different kinds of lipids and with different structures. So first we can define a lipid as a naturally occurring molecule from a plant or animal that is soluble in nonpolar organic solvents, so meaning it is fat soluble. Lipid molecules contain large hydrocarbon portions and some contain a few polar functional groups. But again, lipids are insoluble in polar solvents and soluble in nonpolar solvents. So we're able to classify lipids as waxes. So these are what we call carboxylic acid esters that have long, straight hydrocarbon chains. And we have the triacylglycerols. These are triglycerides. And we'll talk about these in a little while, but these make up fats that are in oils and that are stored in our body and so on. Glycerophospholipids. These are triesters of glycerol. We'll talk about glycerol in a few minutes. And they contain charged phosphate diester groups. These are abundant in the cell membrane. And what they do is help control the flow of molecules into and out of the cells. Remember, nutrients need to get into the cells and waste needs to leave the cell. Some other classes of lipids, sphingomyelins. And these are amides that are derived from amino alcohols, and these contain a charged phosphate diester group. These are important for the structure of the cell membrane and very abundant in nerve cell membranes. We then have the glycolipids. These are again amides, and these are derived from sphingosine, and they contain polar carbohydrate groups. We then have the steroids, and steroids serve as hormones. Cholesterol contributes to the structure of cell membranes, and we'll talk about the link between steroids and cholesterol in a little bit. And then we have the Eicosanoids, and these are carboxylic acids, and we refer to them as intracellular chemical messengers. Let's take a look first at fatty acids. It turns out that naturally occurring fats and oils are what we call triesters, and this should say formed, and they are formed between glycerol and fatty acids. Remember, glycerol is a trialcohol. So let's first go ahead and define a saturated fatty acid. This is a long chain carboxylic acid that only contains carbon-carbon single bonds. Remember, saturated carbon-carbon single bonds. So this here is palmitic acid, and this is a saturated fatty acid. We also have unsaturated fatty acids, and these are long-chain carboxylic acids that contain one or more carbon-carbon double bonds. So linolenic acid is an example of that. We see these carbon-carbon double bonds within the structure. And by the way, this is a cis fatty acid. And remember, we look at the double bonded carbons. If each of the carbons has two different groups bonded, then it can show cis trans isomerism. In this case here, the hydrogens are on the same side, so that makes it a cis. Let's go ahead and define a triacylglycerol or a triglyceride, so we can call them by both. I'll refer to them as triglycerides, sometimes as triacylglycerols. So triacylglycerols are triesters of glycerol with three fatty acids. So here is glycerol. Again, you're familiar with this. This is that trialcohol. This would represent a fatty acid. You have this carboxyl group and then this very long hydrocarbon chain. These fatty acids can be saturated or unsaturated. It doesn't matter. You go ahead and react one molecule of glycerol with three fatty acids. Again, they can be saturated, unsaturated. They can be the same. They can be different. And what we end up with is this triacylglycerol or triglyceride. And remember, a carboxylic acid, right, and an alcohol will form this ester-like compound. So here we have our triester. Okay, so we have triester of glycerol with 3 fatty acids. So it turns out that fats or oils from natural sources are complex mixtures of different triglycerides. Vegetable oils consist mostly of unsaturated fatty acids, whereas animal fats consist mostly of saturated fatty acids. Remember, we're told we should eat more unsaturated fats than saturated fats. I'm just going to mention this right now, that linoleic and linolenic acids are essential in the diet. What that means is our body cannot synthesize them, so we must get them from dietary sources. And we'll talk about these 2 fatty acids in a little while. Okay, so let's take a look at the properties of fats and oils. So the more double bonds that are present in that hydrocarbon chain, the lower the melting point. So when we refer to saturated fats, we know that the melting points will increase with the number of carbon atoms in that chain. So triglycerides or triacylglycerols, more unsaturated acyl groups present, the lower the melting point. So the more unsaturated groups, the lower the melting point. So oils obviously are lower melting. They're liquids at room temperatures. Fats, like lard, are higher melting. Butter, lard, they're solids at room temperature. So we can define an oil as a mixture of triacylglycerols that is a liquid because it contains a large proportion of unsaturated fatty acids. A fat, again, it's also a mixture of triacylglycerols, but a fat is solid because it contains a high proportion of saturated fatty acids. So let's take a look here. Here we have stearic acid. This is an 18-carbon saturated fatty acid. Okay, and so that means there are no carbon-carbon double bonds. Now, on the other hand, we have this linoleic acid, which is an 18-carbon unsaturated fatty acid. There are cis double bonds. So the more double bonds, the more difficult it is to solidify. So that's why oils, because they contain those unsaturated fatty acids, are liquids. Think about what happens when there are no double bonds in that hydrocarbon chain. These molecules compact more tightly together. That means that the dispersion forces between the molecules are much stronger. Therefore, the animal fats tend to be solids at room temperature. When we have double bonds, these molecules cannot pack as tightly together. Therefore, the dispersion forces are weaker between the molecules, and this would result in an oil. Okay? Remember, the stronger the dispersion forces, the tighter those molecules are held together, okay, and the higher the melting points. With the double bonds in the carbon chain, because these molecules cannot pack as tightly together, fewer dispersion, or I should say weaker dispersion forces, meaning the melting points would be much lower. Okay, some properties of triacylglycerols. They're uncharged, nonpolar molecules. They're what we call hydrophobic, meaning water-fearing. So they don't dissolve in polar solvents. So hydro, water, so water-fearing. They're not going to dissolve in water. Triacylglycerols are a major component of our diet, and They are also depots for energy storage, which we'll see a little bit later. Some simple triacylglycerols that we're all familiar with, butter, lard. Butter and lard are saturated, okay? Those are going to be solids at room temperature. Then we have our olive oil, peanut oil, and our other oils. We can react triglycerides, or triacylglycerols, with hydrogen. We would also need a catalyst, usually a metal catalyst. And just like our reaction of alkenes, a hydrogenation reaction where we add hydrogen to the double bonded carbons, we can do the same thing. And this is how they make margarines. Remember, we've been told now that margarines are not good for us because they contain trans fats, okay? And this is what happens. Hydrogen is added to the double bonds, and we end up with a hydrogenated oil, okay? And we can actually control the extent of hydrogenation, and that's just going to depend on how many double bonds and the location of those double bonds in the original oil. So they take an oil, it undergoes a hydrogenation reaction, and you end up with a more solidified product like Crisco or margarine. And we know that if you look on the ingredient list of Crisco or margarine, it'll say partially hydrogenated oil. And we know that we should stay away from anything that is partially hydrogenated. That's because of those trans fats. Those are what are bad for us. In fact, now some new studies are showing that maybe saturated fats are not all bad, but these trans fats definitely are. Let's take a look at what happens when we hydrolyze a triacylglycerol. So basically, we can react them. They react with water to form carboxylic acid, salts, and alcohols. So in the body, the reaction is catalyzed. We haven't talked about catalysis yet, but it's catalyzed by these enzymes called hydrolysis. And this is the first reaction in the digestion of dietary fats and oils. So it turns out that in order to produce a soap, hydrolysis of fats and oils is carried out by a strong base like sodium hydroxide. And we call this process saponification. For example, you could do this in the lab. You might have done it in your lab. Take a fat or an oil, and again, we have water and a strong base like sodium hydroxide. And look what happens. In this reaction, we end up with the glycerol and 3 fatty acid salts. Notice these are the salts, okay? This is the soap. Well, again, you've probably done this reaction in the lab, so I won't go into any more detail, but you should know what saponification is. How does soaps work? Well, let's take a look at one of these, right? What we have here is a fatty acid salt. But remember this R group, that's a very long hydrocarbon chain, okay? So we use soap to wash our dishes. Again, soaps have the polar portion, okay? So you have that carboxyl portion, but also the very long hydrocarbon chain. And what'll happen is these spherical clusters called micelles will form, okay? And what's interesting about the micelles is the way they form is so that their hydrophobic ends are in the center and they're hydrophilic. Remember, hydrophilic is water-loving or water-soluble. So you have the hydrophobic ends in the center. This is the micelle right here, or a representation of one. Remember, the hydrophobic portions are going to be the long hydrocarbon chains, and then the hydrophilic ends, those carboxyl groups, those polar carboxyl groups, are on the surface. You have those hydrocarbon chains that are clustered together at the centers of the micelles, and what will happen is you have greasy dirt, right? You're washing dishes. That greasy dirt is going to be dissolved in the center where those where the hydrophobic portion is, and then It's going to be carried away through the water, right? Because the surface of the micelle is hydrophilic. So that's just a brief description of how soaps form micelles and are able to cut through grease. Turns out the soaps and detergents are similar in their chemical properties. Soaps are produced from natural products, whereas detergents are synthetic. When we say synthetic, we mean man-made. When a soap is used in hard water, what we have are calcium and magnesium ions. What will happen is these ions will encounter the anions of the fatty acid, and they form good old soap scum. So what is the result? Well, we waste soap. We have soap residue in clothing. We have soap scum in bathtubs, which is almost impossible to clean, also in showers, and our drains clog up. Detergents don't do this. They don't cause soap scum like soaps do. But we do classify both detergents and soaps as surfactants, and that's because both of them form those micelles around greasy dirt. That's how our clothes get clean, our bodies get clean, our dishes get clean. So both soaps and detergents have hydrophilic heads and hydrophobic tails. The hydrophilic heads, they can either be ionic, cationic, or non-ionic. By ionic, we mean negatively charged. By cationic, we mean positively charged. And non-ionic means neutral or zero charge. It turns out that anionic surfactants are used in laundry products. Cationic surfactants are used in fabric softeners and shampoos and different disinfectant soaps. And non-ionic surfactants are low sudsing, and they're effective cleaners at very low temperatures. Cascade dishwashing soap is a good example. Okay, so the dishwashing detergents that we use in dishwashers, also our high energy washers, use low sudsing detergents. Okay, so just a little bit about soaps. The next type of lipid is a wax. And our simplest fatty acid esters are waxes. What is a wax? It's a mixture of monoesters of long chain acids with long chain alcohols. So the acids usually have an even number of carbons, anywhere from 16 to 36. The alcohols also have an even number of carbons, 24 to 36. Here we have the structure of beeswax. And this is formed from tricontinyl and palmitic acids, so an alcohol and a fatty acid. So this is it here. This would be the long chain alcohol portion, and this would be due to the long chain acid. So waxes, very nonpolar. You've all seen the commercials with the birds being cleaned off with Dawn detergent. So whenever there's an oil spill, it's imperative that oil is washed off. And they use Dawn detergent. It's a very good detergent, actually. It cleans, there are a lot of uses for Dawn. But anyway, the reason why is these buoyant birds have a wax coating, a waxy-like coating on their feathers. And remember, likes dissolve likes. So waxes are nonpolar, right? Oil is nonpolar. So therefore, that waxy coating on the wings of the bird will dissolve in the oil. That's why we need to get the oil off, because without that coating, those birds are unable to remain buoyant in the water, and they will perish. The next classification, we have glycerophospholipids, and also known as phosphoglycerides. And basically what we have, as far as the structure goes, we have a lipid in which glycerol is linked by ester bonds to two fatty acids. One of them is usually saturated. The other one is usually unsaturated. We have a phosphate here, so that's linked to a phosphate. And then we have another ester-like bond to an amino alcohol or some other alcohol. So if you were to hydrolyze this, we would have the fatty acids, the glycerol, the phosphate, and the amino alcohol or the other alcohol. These are found in cell membranes, and they help to control the flow of molecules into and out of the cells. Again, they're most abundant in the cell membrane. You're starting to see that a lot of these lipids all have these long hydrocarbon tails and also a polar part to the molecule. Okay, so glycerol phospholipids are what we call emulsifying agents. So phosphatidylcholine, that's the basic structure of lecithins. So lecithin is actually found in egg yolks, and it's also in chocolate bars and other foods. And what it does, it keeps the oils from separating out. So, for example, when you can make mayonnaise using egg yolks and oil, and the oil is not soluble in the yolk, but basically if you use your food processor and you add the oil slowly, what you end up with is a mayonnaise. Okay, so it's the lecithin in the egg yolk that emulsifies those oil droplets in the mayonnaise. Think about a chocolate bar. There's also lecithin in chocolate bars. And think about how unappetizing a chocolate bar would look without it, because what you'd have is you'd have this mess where all this oil is separated out. So it's the lecithin that keeps the oil from separating out, and that's why we just love chocolate. Maybe it would be a good idea to keep the lecithin out. I might not eat as much chocolate as what I do. So again, phosphatidylcholine, this is a phospholipid in the lecithin. The reason they're good emulsifying agents is because of their combination of the hydrophobic tails and their hydrophilic head groups. Okay, so the polar groups here. So basically they can surround the drops of the nonpolar liquids like the oils and hold them in suspension in water. Take a look at your chocolate bar, and you'll see that lecithin is listed as an ingredient. And that's usually in chocolate bars, it's obtained from soybean oil. It's just going to keep the oil from separating out from that chocolate bar. The next class of lipid we're going to look at is a sphingolipid. And these are what we call phosphate esters. Remember, we talked about that. We can have a phosphorus atom. That'll be instead of a carbon atom, right? And so you have that phosphorus that's double bonded to an oxygen, then single bonded to another oxygen to form that phosphate ester group. So this lipid is derived from an amino alcohol that's called sphingosine. And this here is that alcohol. So this is found in sphingolipids. And sphingolipids are important as far as cell membrane structure, and they're also abundant in nerve cell membranes. In fact, sphingomyelins are the major components of coating around the nerve fibers called myelin sheaths. And we find them in large quantities in the brain tissue. The alcohol, the sphingosine, it provides one of the two hydrophobic hydrocarbon tails. Okay, so this one here comes from the sphingosine, and the second hydrocarbon tail comes from a fatty acid. And that's connected, as we see, by this amide link to the amino group in the sphingosine. We have the typical two hydrocarbon tails. Also, there's this phosphate group, and then we have this group right here. The next would be the glycolipids, and these are actually derived from sphingosine. And instead of a phosphate group, what they have is a carbohydrate group, okay? And we see that this carbohydrate group is bonded at carbon 1. So we don't have the phosphate group. These reside in the cell membranes, and we'll see a little bit later when we get to metabolism that these carbohydrate segments will extend into the fluid that surrounds the cell. So these function as cell receptors, and we'll talk about what those are a little bit later. We classify a glycolipid molecule as a cerebroside, and cerebrosides, which contain a single monosaccharide, are partially abundant in nerve cell membranes in the brain, and in that case the monosaccharide is D-galactose. This one here is shown with D-galactose. They're also found in other cell membranes, and in those cases the sugar unit is a glucose. Let's talk a little bit about the cell membranes. We already know now that they contain phospholipids and glycolipids. And our cell membranes, animal cell membranes, contain cholesterol. This is the structure of cholesterol right here. Notice these 4 rings here. Cholesterol is the most abundant animal steroid, and it not only serves as a component of the cell membranes, but also it is the starting material for the synthesis of steroids. So all of the steroids have this basic 4-ring structure, and the other groups coming off of the 4 rings are going to be different depending on the type of steroid. So cholesterol, some of our cholesterol is obtained from the diet. Cholesterol is synthesized in the liver. Again, it's the most abundant animal steroid. The body of a 60 kilogram person contains about 175 grams of cholesterol, and it serves 2 important functions. One, it's a component of cell membranes, and again, it is the starting material for the synthesis of all other steroids. Cholesterol also contributes to heart disease because of its presence in arterial plaque. Even on a very strict no cholesterol diet, an adult will usually manufacture about 800 milligrams a day of cholesterol. If we go back here to the structure, we only see this little polar group, right? The rest of this is mainly carbon and hydrogen. So we see that cholesterol is a hydrophobic molecule. And because of that 4-ring structure, that means that it is a very rigid molecule. Cholesterol is very important to maintain the integrity of the cell membrane structure. Even though people are told to limit their cholesterol intake, again, we do manufacture our own cholesterol, and it is definitely an important component when it comes to the cells. What we're going to talk about here is the structure of cell membranes. Okay, so phospholipids provide the basic structure of cell membranes, and they aggregate in a closed sheet-like double leaflet structure that we call, the lipid bilayer. This here would represent our lipid bilayer. The bilayer is formed by 2 parallel layers of lipids, and they're oriented so that the ionic head groups are exposed to aqueous medium on either side of the bilayer. And then we have those nonpolar tails, and what they do is cluster together. We see them here, here, and here. They cluster together in the middle of the bilayer, and they interact together, and they avoid water. They're hydrophobic. So we refer to each half of the bilayer as a leaflet. Okay? So the bilayer is a great place for the arrangement of phospholipids. It's highly ordered and stable, but even though it's highly ordered and stable, it's still very flexible. So if you were to take some phospholipids and vigorously shake them with water, then what we would form is what we call liposomes. So what these are small spherical vessels that have a lipid bilayer surrounding an aqueous center. The overall structure of the cell membrane is represented by what we call the fluid mosaic model. Okay. And again, we describe it as fluid because it's not rigid and molecules can move around within it. And we describe it as a mosaic because it contains many kinds of molecules. So here we can see the components of the cell membrane in this figure right here. We see the different components. It turns out that 20% or more of the mass of the membrane consists of protein molecules, and many of them are what we call glycoproteins. Okay, so right here we see some glycoproteins. What we have are peripheral proteins, and these peripheral proteins, which I am looking for right here, the peripheral proteins, here we go, we have one right here. They're associated with just one face of the bilayer, so with one leaflet, if you will. And they're held within the membrane by non-covalent interactions with the hydrophobic lipid tails or the hydrophilic head groups. The integral proteins, and here we have an example of an integral protein, they extend completely through the cell membrane. And they're basically anchored by hydrophobic regions, and these extend through the bilayer. Sometimes the hydrophobic amino acid chain can twist in and out, as we see here. It can twist in and out of the membrane many times before ending on the exterior of the membrane. And they will usually have a hydrophilic sugar group. The carbohydrate parts of the glycoproteins and glycolipids help mediate the interactions of the cell with any outside agent. So some of the integral proteins, like this one here, this represents one, will form a channel. And these channels will allow specific molecules or even ions either to enter the cell or leave the cell. Again, we stated that the bilayer membrane is fluid rather than rigid, and that's good because it's not easily ruptured. The lipids in the bilayer will simply flow back together to repair any small tear, hole, or puncture. You can think of this as being similar when you're cooking. So you have a thin layer of oil or butter. It might lie on top of water in a cooking pot. You can puncture the film and it can break, but then it immediately flows back together if you leave it alone. The great thing about membrane fluidity is that proteins are able to move within the cell membrane. We have lipoprotein receptors, which are glycoproteins, and these interact with lipoproteins in the extracellular fluid. They move sideways within the membrane to form clusters of receptors on the cell surface. Another consequence of the bilayer fluidity are that small nonpolar molecules can easily enter the cell through the membrane. And some individual lipid or protein molecules can easily diffuse rapidly from place to place within the membrane. The fluidity of the membrane is going to vary with the relative amounts of saturated and unsaturated fatty acids that are in those glycerophospholipids. So, for example, in reindeer, the membranes of cells near the hooves contain a higher proportion of unsaturated fatty acid chains than in other cells. Well, what do we know about that? Well, we know the unsaturated fatty acid chains do not pack as tightly together. So what you have is then a membrane that remains fluid while the reindeer stand in the snow. We're going to talk a little more about the membrane later, but now, it's just good that you know about the integral proteins, the peripheral proteins, and how some of these glycoproteins are able to extend through the membrane. Some are only on one face of the membrane. Some form channels to allow molecules in and out of the cell, and so on. Do note that cholesterol, okay, we see representations of the cholesterol, and again, because of that very rigid ring structure, this maintains the structural integrity of the cell membrane. Okay, so here I just talk about the fluid mosaic model. This means molecules can move about within it. Contains many molecules. We already said the membrane is composed of a fluid-like phospholipid layer. Glycolipids and cholesterol. 20% of the mass of the membrane consists of protein molecules or glycoproteins. And again, we said peripheral proteins are associated with one face of the bilayer, and that they're held in the membrane by non-covalent interactions with hydrophobic tails or hydrophilic head groups. Integral proteins, again, extend completely through the cell membrane. They're anchored by hydrophobic regions that extend through the bilayer. So, as I said before, the hydrophobic amino acid chain we haven't talked about amino acids yet, but it can twist in and out of the membrane many times, okay? And then usually ends on the exterior of the membrane with a hydrophilic sugar. Remember, sugars, those monosaccharides, are very hydrophilic because of those OH groups. So the integral proteins mediate interactions with other molecules, proteins, enzymes, and so on. Okay, we already talked about the bilayer being fluid and not easily ruptured. As I said, small nonpolar molecules can enter directly through the membrane. Some lipid or protein molecules can diffuse quickly from location to location within that membrane. The function of the cell membrane allows passage of small molecules and ions into and out of the cell. There are two types of transport. Passive transport, and this is movement of a substance across a cell membrane without the use of any energy. And this is from a region of higher concentration to a region of lower concentration. And that's how it would work in nature, which is why no energy would be required. And then what we have is active transport. And this is movement of a substance across a cell membrane, but with the assistance of energy. So, for example, later on, we're going to talk about ATP, which is the body's energy molecule. So we'll talk about these types of transport a little bit later, simple diffusion and facilitated transport, a little bit later when we get to metabolism. The next class of lipids I want to talk about are the eicosanoids. These include leukotrienes and prostaglandins. An eicosanoid is a lipid derived from a 20-carbon unsaturated carboxylic acid, synthesized in the body as a response to some external stimuli. They function as short-lived chemical messengers, so similar to hormones, and these act near the point of synthesis, unlike hormones. We refer to them as local hormones. Prostaglandins and leukotrienes are synthesized from the 20-carbon unsaturated fatty acid called arachidonic acid. And arachidonic acid is synthesized from linolenic acid. Remember, we said that linolenic acid was essential, meaning that we don't synthesize it in the body, so it has to be obtained from the diet. Prostaglandins contain a 5-membered ring. So they're carboxylic acids. We see that they contain the a 5-membered ring here. And I'm not even going to go into their biological activities, but they do have a very wide range of biological activities. In fact, we talked about prostaglandins in a previous chapter. Prostaglandins are responsible for pain and swelling that's caused by inflammation. So we talked about, we take, for example, aspirin for inflammation, pain, and swelling. And so what that does, it inhibits the synthesis of the prostaglandins. So that way we feel pain relief and hopefully some relief with the swelling. Prostaglandins are also known to lower blood pressure, and they also influence platelet aggregation during blood clotting. They can stimulate uterine contractions. And again, in a previous chapter, we talked about how they work with pain relievers. The leukotrienes, it turns out when these are released, an asthma attack or allergic reaction can be triggered, as well as inflammation. So some of the newer asthma drugs now, they inhibit the synthesis of leukotrienes in the body, and which of course helps to treat the asthma. That's it for lipids. So know the different classes of lipids and basically what I talked about in this video, and you should be fine. If you have any questions, let me know and I'll be more than happy to answer them. So everybody have a great day.